Hello and welcome to today's video, where we'll be discussing the neurobiology of Down syndrome and its associated effects on human functioning. We will begin with a short introduction outlining the different types, characteristics, and effects of Down syndrome, before discussing the differences in neurotransmission, neuroplasticity, and neurodegeneration. Down syndrome is the most common example of a neurogenetic aneuploid disorder leading to mental retardation. In most cases, Down syndrome results from an extra copy of human chromosome 21 producing deregulated gene expression in the brain that gives rise to subnormal intellectual functioning. There are three types. These are trisomy 21, where there are three separate copies of chromosome 21 resulting from random error during cell division. Translocation Down syndrome. This is where an extra part or whole extra chromosome 21 is present, but attached to a different chromosome rather than being separate. Mosaic Down syndrome. In this type, some cells have three copies of chromosome 21, while others have two. Common characteristics of Down syndrome include physical alterations such as a flattened face, slanted eyes, and smaller hands, feet, and ears. The cognitive effects often relate to learning and memory. The brain is much smaller than normal, and the shape is rounded, short, and has a relatively narrow anterior to posterior diameter. Now we will move on to discuss the differences in neurotransmission for Down syndrome people. Multiple physical changes occur in the neurons of a Down's brain. There is a reduction in the overall number of neurons in the brain, especially in the temporal lobe. Morphological changes occur to the dendritic spines, with one unusually long spine, one shorter spine, and a reduction in the overall number of spines common. Further to this, there is a precocious cessation in the development of dendritic spines of visual cortical neurons. Down syndrome patients suffer from excessive inhibition, which leads to an imbalance that compromises neural circuit function. This overinhibition is usually related to GABAergic transmission in the brain, which disrupts the optimal excitatory-inhibitory synaptic balance, which depolarizes GABA transmission. Glutamatergic transmission is also impaired. This dysfunctionality of the brain accounts for impairments in synaptic plasticity and learning and memory deficits in Down's people, as well as behavioral disability. We will now progress to talk about the impairments in neuroplasticity in the Down syndrome brain. Neuroplasticity is an intrinsic adaptive ability of the nervous system in response to the changing milieu, continuous neural activity, learning, and experience. Basically, neuroplasticity accounts for learning and memory, capability of the nervous system, and is also responsible for regaining lost abilities of the damaged neuronal system through structural and functional rehabilitation. The main structure of the brain with impaired neuroplasticity in Down's patients is the hippocampus. Impairments in neuroplasticity in Down syndrome people are due to the presence of excessive inhibition in the brain. This imbalance prevents the processes of long-term depression and activity-dependent reduction in the efficacy of neuronal synapses, and long-term potentiation, persistent strengthening of synapses, from functioning aptly. The Down's brain is subjected to more long-term depression. Essentially, the plasticity of the brain is controlled by the efficiency of the synapses, with intracellular calcium levels the main trigger for a cascade of intracellular messengers that lead to the induction and maintenance of synaptic plasticity. Down syndrome people have a high percentage of primitive synaptic contacts and reduced synaptic parameters. This means that the development of the hindbrain in early life is inhibited, as the cells of primitive synaptic contacts are mainly responsible for the development of the forebrain. In addition, reduced synaptogenesis, which is the long-term developmental process involving synapse formation, refinement, and elimination, and is crucial for the establishment of the neuronal network and precision of brain circuitry, and alterations in the synaptic morphological development in the Down's brain are significant contributing factors to the deficits in neuroplasticity. Significantly impaired neuroplasticity, such as that present in the Down syndrome brain, results in decreases in cognitive functioning and neuronal maturation. The combination of these factors leads to delays in physical and mental development. However, recent research has provided hope for these individuals. Experience-dependent cognitive training, such as employing visual learning for Down syndrome individuals with hearing impairments, has begun to show improvements in neuronal and synaptic plasticity, and therefore reduce the disabling cognitive and intellectual effects of Down syndrome. To finish today's video, we will be discussing neurodegeneration. Neurodegeneration is a common issue associated with Down syndrome. This relates to the progressive loss of neurons and therefore a decrease in brain functionality. It is said to be caused by restricted oxidative transport to the brain and reduced neuronal repair activity. Neurodegeneration and the high risk to develop amyloid neuropathology are part of the neurobiological alterations occurring in individuals with Down syndrome in adult age. Several neuroimaging researchers have long showed that adult Down syndrome persons display typical brain alterations occurring in Alzheimer's disease. After age 40, the concentration of amyloid proteins in the brain increases markedly, which is associated with the formation of neurofibrillary tangles. combined with increasing amounts of plaques, this causes the death of neurons in the brain. So what are the precursors to this neurodegeneration? Mitochondrial dysfunction is noted to be the reason for the decreased oxidation of the brain. Additionally, there are a number of genes present on chromosome 21 that have been implicated in Alzheimer's disease, including superoxide dismutase 1 and beta secretase 1. Therefore, the presence of an extra copy of this chromosome directly leads to an increased risk of Alzheimer's disease. Evaluation of brain 3D anatomical images of patients aged 40 years or older revealed a relationship between cognitive deterioration and severity of specific anatomical changes, such as in the basal forebrain and hippocampus. Finally, the early signs of dementia in people with Down syndrome might be a result of dysfunction of the frontal lobe and hippocampus, areas in which amyloid first accumulates during the early stages of dementia. In addition to the previous points, low levels of noradrenaline and impairment of noradrenergic transmission correlated with the occurrence of neurodegeneration and dementia in adults with Down syndrome. Indeed, detection of low levels of noradrenaline was shown to increase the risk of developing dementia in Down syndrome adults by tenfold. That concludes today's video. Thank you for watching.